All right, I'd like to introduce our first speaker for the session. Christopher Boccia is from Queen's University, and he's going to be presenting on optiMander, a classifier neural network for individual identification in salamander species with low inter-individual visual differentiability.
All right, thank you, and apologies for the very difficult title. All right, so yeah, I'm going to be talking about attempting to make a neural network for identifying individual salamanders in an endangered salamander population. All right, so the salamanders that I'm working with are Jefferson salamanders. So they're ambistomatid, they are mostly subterranean, and they come out in the early spring and breed in vernal pools. This is just a nice one here. And so the study that I'm working with, their objectives are to assess breeding adult numbers at a specific population area in Southern Ontario, identify current threats to the salamanders at the site, and kind of just to form a comprehensive conservation and rehabilitation plan. So I'm not the lead on this project. Meredith Swartout, if you're interested in all this stuff, is going to be giving a great talk later this afternoon in the Charlie White Theater. I encourage you all to check it out. So I'm just going to go over some of the details very quickly, since she'll be talking about them in a lot more depth. And so what I'm going to be focusing on is assessing breeding adult numbers. And so to be able to do this, we have to be able to differentiate one salamander from another. And in Jefferson salamanders, that's a little bit tricky because they look really similar. So the dorsum is usually patternless. So unlike a lot of other ambistomatids where we have rings or spots to be able to differentiate them, we don't really have those to work with on the surface anyway. And as you can see with these two here, you wouldn't necessarily be able to tell them apart very easily. So the usual ways that people have gone about this with salamanders like this in the past for individual identification are toe clipping, visible implant elastomer tagging, and pit tagging. So for this project, we've been doing VIE tagging because it's the least invasive of these. Toe clipping has been shown to have negative survival consequences, and pit tags are fairly large, and it has been done for some movement studies, but you don't necessarily want to do it to every individual in a population. So VIE is really quite a good safe technique. There's a lot of papers and studies that have managed to do this really well and used it for individual identification. But there are some downsides, is that it still requires a lot of handling and pretty expert injection technique. And so it also takes quite a long time when you're marking a big population because you have to do multiple marks that are of multiple colors, as you can see up there with that salamander up here. And so overall, it would be better if we had a simpler non-invasive approach, so limited handling time. And that's especially true as this is an explosive breeding species. So in the spring, sometimes we might get 300 individuals in a night. That's a lot to have to do with VIE tagging. So with all this in mind, while we were working on the VIE project, we noticed that most of the Jefferson salamanders in our population had blue chin frecklings. These are 3 examples here of individuals with that. And so these freckling patterns appear to be unique to each individual. We don't really have longitudinal data on this yet since we've only done 1 field season, but as far as we can tell, they haven't really been changing. There's some possibility that this is only in unisexual Jefferson salamanders. So this population has a high proportion of unisexuals, and those are hybrids, so they have some blue-spotted salamander genetic contributions, and so that might be the cause of these spots below the heads. And interestingly, it looks like the pattern shows up when they're metamorphs. So we've been kind of tracking them since they've come out of the ponds. And initially, they don't seem to have pattern, but more recently, we caught one under a cover board. And you can see there's just there's a spot right there. So we're not sure if it'll continue like that to adulthood, if more will be added, but it'll be interesting for us to keep an eye on that in the future. So yeah, with this, I thought, maybe if these patterns are consistent, we could do a neural network approach for identification. So we wouldn't have to do the VIE long term. And so at the time, I thought this might be a really novel idea, but apparently in the last 5 or 6 years, everyone's been starting to look into neural networks for salamanders. There's a bunch of other programs that have been attempted. So people have done it for looking at color morphs and redbacks. There's been some work on newts as well, and Japanese giant salamanders. And of course, there's the long term, or the program that's been out for a while, I3S, that works on salamander spots. So in terms of what we have, our data set, we have about 2,500 images from about 700 individual captures. We do have some repeat individuals, which is great for training our model and testing it out. Unfortunately, this was something that we kind of tacked on last minute. So we have some really great quality photos, but then in a lot of cases, we were just taking these with people's phones, and not everyone had the same photography skills. So we do unfortunately have a lot of poor quality photos that are still usable, but might make things tricky. So step one is we want to be able to have the model identify a salamander chin from a photo. And so this is mostly to ensure that there's not too much noise going into our model. So as you can see here, this is an example of a photo that we have, and it's got a scale, like a ruler for scale here. But if you have any experience with neural network methods, they'll pretty much seize on anything. So if, for example, if all the pictures of this individual are right next to this particular set of marks on the ruler, the neural network might pick that up as opposed to the chin pattern. And so in the future, it'd be like, well, it can't be this individual because the ruler's not there. And similarly, if you look down here, this is one of the VIE marks. So that could also be picked up if we just ran this image through the neural network. And so that would kind of eliminate the point, as we would then need to have the VIE marks to be able to identify the individual. So anyway, I labeled about 200 of them with these bounding boxes here. It's a little bit hard to see, but it's in pink there. And then I used that to train a neural network to automatically generate these bounding boxes on all the other images. And it worked pretty well. We did have some images crop in that were identified the wrong thing, but in general, it was able to segment out the chin. And the nice thing is with this is we'll be able to run all these images through in terms of training, and that should help for future runs through of the model. One thing we did notice was some of our images were taken on sort of unusual backgrounds. So mostly we fortunately had a light background, but anything that didn't have that, the model struggled with that. So we'll have to have more training cases with that in the future. And yeah, so that's kind of the first step. So we managed to be able to identify where the chin is in the photo. But of course, there's still some background noise. So you can still see up there that even with the bounding box there, we're still going to get a little bit of the ruler in. So what we want to be able to do is just have just the head and only the head. And so to do that, I used the bounding boxes and I ran segment anything, which is a model implemented by Facebook actually in Python. And so that was able to then select just the chin itself. And then that way we were able to mask the image. So we get chin and no other noise around it. All right, so at long last, actually trying to identify individuals. So I preliminarily tried out SIFT, which is the same underlying algorithm that's used in I3S. And so the way it works is it identifies scale and rotation invariant features. So when you have your photos, sometimes they're obviously not going to be in exactly the same orientation. So you want to be able to still account for that. So that's what SIFT does. It identifies key points. And then you try and use those to figure out shared key points. And you can determine homology by the distance between those points. So initially, it looked like this was going to work really well. So these are two images of the same individual here. And you can see SIFT is picking up the same points and identifying them as being relatively close to each other, which is great. However, we found with a lot of our images, so this is the green dots here are the key points that SIFT identified in the image. It was able to pick up the dots as key points, but when we actually ran the feature matching part of the algorithm, it wasn't able to identify that those were shared. So these are the same two images, and these are the points that were identified as being shared. And some of these are kind of farther apart than we would like, and that kind of means that the overall distances that we get for homology aren't too accurate. So we're not totally sure why this is happening. My current guess is just that often, the salamanders are wet when we're photographing them, so there's a lot of reflectance from the lights we're using. So maybe that's part of it, or maybe the spots are hard for SIFT to differentiate. So we did try some thresholding to try and get around this, but that kind of opens up an entirely different can of worms and didn't add too much to our success. So I think SIFT could totally work for this, but I think we would have to define reference points and then actually indicate to it which dots to look at, which we could do, but the whole point of this is to kind of have a fast and effective method of identification. So that's not really what we were hoping to have work. So instead, what I've been using is now a convolutional neural network approach. So I won't go too deeply into the methods. If anyone's interested, you can chat with me after. But the idea here is it was first, the code that we're specifically using right now was used for giraffe sides originally, which is a fun use. And the nice thing with the neural network approach is that we get to give it both positive and negative cases. So with SIFT, it just identifies key points, and then that's what's used. But with the CNN, we can tell it, okay, this is that salamander, and this other one is not. And so that way the points and the weights that we end up getting can be a lot more specific and a lot better. And overall, I only have had time to run this through for a small subset of our individuals so far, but the results seem to be pretty good. So what we're mostly looking for here on this plot is we have distances. So this the neural network essentially estimates, similar to SIFT, a distance between each image. And so in this case, these are the test images on this side. And down here are the reference individuals. And so there's a few images for each. So what we want to see is the darkest point on the row matching up with some of the photos of that same individual. And for pretty much all of them on the line in the diagonal there, that's what we see, with one exception over here. This one here was identified as a different individual. But yeah, so out of this test set, about 9 or 10 images were identified as closest to the correct individual. And even for the one here that it got wrong, the correct individual was within the top 5. All right, so yeah, given all that, eventually once we run it on everything and we're more confident in our results, I'm hoping to release all this as a user-friendly code package that people can use for their systems. And I'm also really hoping to implement it on site next year when we're doing the next stage of the Salamander project. And that'll kind of give us a way to ground truth it. So we'll be able to scan the chins. It'll give us what the expected VIE code is, and then we can flip the Salamander over, check the VIE code, and and then just make sure that the model is actually giving us something accurate. And yeah, we can get an idea of its success in the field that way. And yeah, long term, I'm hoping that that will mean that we can just use just photos to keep tabs on this population, and there won't be a need for VIE. And yeah, I'm also hoping to eventually turn this into an app. So there's a setup for Android for running some of these neural network models. I'm hoping to make use of that. And then that way you could just do this all using a phone database and do it right in the field. And yeah, hopefully that'll, at some point I can also maybe try and generalize it to other taxa. Obviously not everything is going to have a chin pattern of interest. All right, and yeah, with that, I'd just like to acknowledge all the amazing volunteers that we had on this project who came out and helped us with our overnight and all-nighter fieldwork, and in particular, our field assistant, Maddie, who did a lot of the image collation for this project. All right, so again, I'll take any questions.
How do you hold your salamanders still and in the constant position for those photos? The reason I asked that we've been trying to use pattern mapping for a number of species, for snake and for western redback salamanders who also have those freckles. They don't like to be upside down. Sometimes the photos are from an angle, and that has caused us a lot of problems.
Yeah, it's tricky. Like we did kind of a lot of these later ones were taken in the field, and we did kind of manage to get a bit of a setup where you kind of have them put their head between your fingers, and then they don't like it, but you can sort of manage to get them to do that. We are hoping to long term have maybe like a plastic bag or potentially just like a plexiglass setup where we can maybe put them on it and then we can slide the camera underneath. This was just sort of, we were during the field season doing it. We're like, oh, this is a really cool pattern. We should try and get photos. So we didn't really have the best setup this time around, hence why some of the photos aren't great. But yeah, we're hoping, we're thinking maybe the plexiglass where they can be on something where they're flat. Then we can just take the photo facing up. Might be the best approach in the future.
Okay, great. We have time for one more quick question.
Yeah, go for that.
Yeah. Hi. I had a question about how confident are you in the spot patterns across like ontogeny and over time? Do you think these are consistent for the individuals and this would be feasible for monitoring?
We're hoping so. So I mean, we only have one field season's worth of data so far, but we are hoping to do this for like 2 or 3 more years. So hopefully we'll have to get some idea of that. The nice thing is we do have the VIE marks. So if the patterns change, we'll be aware because we're like, oh, it identified it as this individual and the VIE code does not match up at all. So that'll be good to know. We're hoping they don't change, but we'll see. Yeah.
All right. Thank you so much, Christopher. That was an amazing presentation about some very smart stuff, but I don't quite understand, but amazing. So next up, we have Rebecca Golat from Biolinx. She's going to be presenting on needle in a haystack, field methodologies for researching Canada's elusive sharp-tailed snake.
All right. Who's ready for more chin photos? Good morning. My name is Rebecca Golat. I work with Biolinx Environmental Research here in Victoria. For the past 3 years, I've been involved in the sharp-tailed snake project. We're focused on conducting the majority of the field work. And today I'll be talking about some of the field methodologies for finding this elusive species. Ooh. All right, super quick background. Sharp-tailed snakes have a sharp tail. It actually is sharp. This is a really small, slender, cryptic species. So individuals don't usually extend 28 centimeters. They're well camouflaged. Thank you. They're nocturnal. They're semi-fossorial. They're very rarely ever seen out in the open. So when hikers come by and they're like, oh, I saw one of those like on the trail, I was like, I don't think you did. They're designated threatened under SARA. and they're one of the only 2 species of the genus Contea. There has been intensive long-term monitoring conducted at 2 Vancouver Island sites. So the first one is Observatory Hill, which is National Research of Council Canada land, and that started in 2007, and Heels Rifle Range, which is Department of Defense, and started in 2005. This has been a little bit more on and off. This site, it has greater restrictions of when we can visit it, but so we started pit tagging 2022. Both are home to rare Gary Oak and arbutus ecosystems, supporting the sharp-tailed snake alongside many other endangered species. My colleague Kristiina will provide more details on habitat right after my presentation. The main objectives is to understand sharp-tailed snake habitat use, seasonal movement, population size. There's quite a big gap in our knowledge of sharp-tailed snakes, not just in Canada, but in their entire range. This is partly due because they are a cryptic species, but we need a critical habitat designation under the species at risk. Like I said, really small, secretive snake. A lot of conventional methods are impractical because of their size. Even our solution of pit tags, passive integrated transponders, we have to place the tag. The tag cannot be placed subcutaneously because of how slender the snake is. So it actually has to be put into the body cavity, which requires a little surgery. There's 3 main methods that we use for mark recapture. The first, one of the most important, is our artificial cover objects. So these ACOs are black asphalt roofing that we place on our site. So about 300 ACOs at Observatory Hill and 122 at Heels. I think without these, we wouldn't be able to find any snakes. One time I found one under a rock, and then I looked under like 1,000 more rocks and I never found one. So our ACOs are really important. We also observe other reptiles under them, and we note that. So our invasive common wall lizards is probably the most common that we're seeing right now, as well as algae lizards and a couple subspecies of garter snake. Once we have the snake in hand and we've tagged it, the next method will be using pit tag telemetry surveys. So these are using the handheld antenna readers to scan the ground. And the last method is our auto readers, which is longhorn antenna systems. And I'm not going to touch too much on this because Kristiina will be doing a whole presentation on it. But this is kind of a game changer for us because it monitors movement 24 hours a day. This is a site, both these sites are pretty impractical to survey at night. They're really steep. They're pretty dangerous even during the day. I usually fall like once or twice a season. So this year was in front of like one of my new field assistants. Is very embarrassing. This picture does not show how steep it is. This is one of the flatter areas. So when we have a pit tag snake, we will, once it's released back into the wild, we will start scanning from the last known location, which is usually ACO. We'll also spot check other suitable habitat. This is where the needle and haystack comes in because it's a bit of a crapshoot. You're really like finding a pit tag snake away from an ACO is almost more exciting than finding a new snake. This is really like quite exciting. The antennas reach to about 15 to 25 centimeters underneath the ground. So this is helpful when we're feeling particularly fossorial. Our field season runs from early March till mid-June, taking a pause in the hot summer months. And then from mid-September, so we pretty much just started last week, until November. The first photo there in the snow was, I think, at a late snow in April. We actually did find a snake that day. I thought earlier, seeing Carl's photos of the snakes, I thought maybe I had the only snow photo, but he beat me to it. Once we have the snake in hand, we take the normal measurements, and we also take photographs of underneath the chin and tail for identification. We'll take fecal samples, and we'll take blood samples if we take them back to be pit tagged. We take them to our field laboratory, which is Kristiina's shed. And let's go over our pit tag implantation. So first, the snake has to be larger than 3 grams and over 170 millimeters. Like I said, the subcutaneous placement is not possible just because how thin the snake is. So we use a local anesthetic before the incision. And then we use a curved tweezers to find a placement for the pit tag. We use biomark RFID tags. These are usually 8mm tags, but bigger snakes will get a 10mm tag. The 10mm actually are quite easier to identify or to read. So we put it into 10 when we can. After the local anesthetic and the incision, we put the tag in. This is usually when we take a blood sample, which is easier said than done. Sometimes, even though you're making an incision, they don't bleed at all, which is always weird to me. And then we take a little dab of vet glue or like a liquid bandage, and that seals them right up. We monitor them overnight and then release them back to the same ACO the next day. We've noticed no ill effect. We've never had a mortality. They're tough little snakes. And this is pretty critical for monitoring their movements on these study sites. March-in photos. All right, so in conjunction with pit tagging, we use photographs of the chin. And this helps cross-reference these snakes, as well as snakes that are too small to be pit tagged. We can identify them first with chins. So some snakes we identify years before they can actually get a pit tag. We call this our family photo album. And we don't have any models to use. So this is just me going through hand, but for each snake. It doesn't take too long because we don't get too many snakes a year. And it'd be interesting to look into some more modeling, but it would also take my job. So this individual really shows how the photo ID really complements our pit tagging efforts. This snake was initially caught in 2019 and was too small. It was caught again in 2020, still too small. And by 2022, it had grown large enough for a tag. So then once it was tagged, we didn't actually see it in 2023. No observer saw it. Our automated pit tag readers caught it. So we had every kind of method that we have possible this snake went through, and it really extends the life history of these snakes on either side. So our efforts, since 2011, we've tagged 114 snakes. One of those was just last Tuesday. And we've had 66 detections with portable antennas, so excluding the automated readers. Heels rifle range, like I said, we have a way more limited access to this site, so we haven't got as much observer effort. We've had 20 tags, 20 snakes tagged since 2022 and 4 detections. Observatory Hill, we've taken 250 snake photos. So this family photo album is getting quite extensive, and 94 have been re-identified. And so similar with Heels, 105 and 12 re-identified. A surprising result from both the pit tags and the photo IDs is that this species is a lot more long-lived than we first initially thought. All of these bottom photos, so 2012, 2015, 2005, these were all adult snakes when these pictures were taken. So again, Carl was saying that size doesn't equal age, but these are most likely over 3 or 4 years old. So extending these times. So our ages are what? 12, no, 11, 15. And our most notable one here at Mary Hill, this is a site that we don't have any pit tags. This snake was over 20 years old. So in the literature, it claims that the snake's life expectancy is around 9 years old. So this over doubles it. Some other results that we have that we've noticed is that they're relatively sedentary. They usually move less than 50 meters from their last known location, except for one kind of outlier. In 2022, an adult male moved 282 meters, including a 96-meter gain in elevation. I believe this was over a couple of months because a snake that is less than 28 centimeters doesn't move that fast. They show site fidelity, not just to their hibernation sites, which we have 9 known or suspected hibernation sites on Observatory Hill, but also to individual ACOs. So there was an individual that was tagged in 2019, and it wasn't seen for a many number of years. But when it was rediscovered last year, it was under the same ACO. And I think it was something like 5 or 6 years. Exact same spot. We didn't see that snake like between that period of time. So it's a little bit frustrating. Wondering where they go, if they're just a little bit too deep that we can't like read them. And lastly, these are surprisingly long-lived for such a small snake. So we have quite a few snakes over 10 years old. I just want to thank our people and our funders, and all these beautiful photos are by Ryan Wilkes. He came out on field day and got some great photos of them. So I just want to thank all of our people. Thank you so much.
Great presentation, really, really interesting research. And forgive me, I came in a little late if I missed this, but what is your selection process to determine which sharp-tailed snakes you pit tag and which ones you don't?
So it's size.
Just whether or not they physically can. Yeah. Okay, cool.
So over 170 millimeters is usually, and like that's usually a snake that's above 3 grams.
Awesome. Thank you.
All right. Any more quick questions?
Yeah, thanks for your talk. That was fantastic. I'm just wondering, what's the range of those automatic detectors? How far can a snake move until you're not able to detect it anymore? Do you know?
That's Kristiina's talk coming in right
Okay.
after about the automated detectors.
I'll stay tuned.
Yeah. I'm putting on the field trip tomorrow at Observatory Hill, so hopefully I'll see some of you out there. And can't promise anything, but if the weather is looking good, hopefully it keeps raining tonight.
So it's on? Okay. Yeah, great talk. I was curious, you can't put the pit tags in subcutaneously because they're size, but you put them under the muscle tissue then?
Yeah.
Does it form a lump or anything like that? Or does
Actually,
it?
some of the snakes that like I thought, like when I find a new snake and I'm not sure if it's tagged or not, I'll be like, oh, like I sometimes will look for a little scar and I'll be like, oh, this one's probably been tagged and it wasn't, or vice versa. Like that it's pretty much, you can't tell once the scar heals. Sometimes there's no scar at all. So it's really like, you can't tell at all. Like it just really like fits into the muscle, the tissue into the muscle.
Awesome. All right. Thank you so much. Amazing presentation. Super jealous of those of you that get to go on that field trip tomorrow. Hopefully see some of these in person. Next up, we have our travelogue speaker from last night is back for another presentation for us. So Kristiina Ovaska is also from Biolinx, and she's going to be presenting on automated pit tag readers and how they elucidate habitat use by sharp-tailed snakes on Vancouver Island.
Okay, hello. We're going to continue with sharp-tailed snakes. My colleagues and I have been working on sharp-tailed snakes for quite a while now. Two of them are here. Rebecca, you heard from Rebecca, and there's Christian at the back there. You can put your hand up, Christian. Christian will take all the questions. Leonard is not here. He is in Saskatoon attending his mother's 100th birthday, which unfortunately coincided with the CHS meeting, but we can't do nothing about that. So because of the rarity of these snakes, it's been really a struggle to find methods and to get any kind of a quantity of data that we could analyze statistically. This newest method, we've been dabbling with automated pit tag detection methods for a long time, starting with homemade units that functioned so-and-so. And for the last couple of years, we've been, thanks to Eric Gross from from Canadian Wildlife Service, and Leanne, and also Punima initially, and the sharp-tailed snake recovery team, we've been able to get these automated pit tag readers from Biolinx. Oh, I have to make my snake move here.
So fast.
So the objectives, Rebecca went through them. We still, after all these years, we still don't know much or know enough about the seasonal movements and activity patterns of these snakes, and specifically habitat use. So And this is important from the conservation point of view because this information informs critical habitat description under SARA. So the scheduler studies in the recovery strategy stresses the identification of the extent to which adjacent closed canopy forest habitat is used by sharp-tailed snakes. And this is around these warm openings that we know about. So this is required for identification of critical habitat more accurately. We work on the Pacific coast population of sharp-tailed snakes. You just saw this map. This is Observatory Hill, or the site we call Observatory Hill. This is Hill's rifle range. We have another site there in the south, Mary Hill. These are all federal sites and our current study sites, but we don't have any pit tag snakes there. This is what the habitat looks like on Observatory Hill. Some of you may be on the field trip tomorrow with Rebecca, and so you'll get to go there. It's a very, very interesting site. Beautiful habitat, Garry Oak meadows and talus slopes. Here's our design for the automated pit tag readers. So we have open habitat there. I'll see if I can, I'll try to use this. I see everything is double here, but I will see if I can use the pointer. Oh, okay. Okay, it's not pointing, but I can I do this? Can I move? No? Okay, I can't move. So if you look at the top there, that's the open habitat. We know that the snakes occur there. So we have a transect of automated units. We call them tendrils. So it's a porous, it's like a sieve. So they get caught in the automated units. Each of those circular areas is an antenna when they go close to the antennas. And 16 of those run by one reader at the forest edge where we know the snakes occur. Then we have 16 similar transect in the transitional forest, transitional from the opening to the more coniferous forest. And then we go into the closed canopy forest, and this is coniferous forest. And this is what we really want to find out, how much they use this closed canopy forest, if at all. Here we have a somewhat different system. We're using a 20-meter cord antenna. So this is just a line laid out onto the ground. And if the snakes cross it or go close to it, about 25 centimeters or so, it gets red. Its tag gets red. And this function, we have 4 setups, 4 replicas of this setup on Observatory Hill. There you go, 4 replicates. Here's what the habitats look like. This is the opening and the edge habitat. And the transitional forest, you see some Douglas firs there. And deeper into the forest is mainly coniferous forest. In this case, in this replicate, is closed canopies. The difference between transitional and this is more coniferous and further into the forest. But it can also look like in another replicate, it looks like this. It's more Douglas fir forest with the dense understory. These transects of antennas, they controlled by the control box, which houses the reader and batteries. And you can see here all those cords. It's not cordless, unfortunately. So for each of the tendril lines, there's 16 cords, and we can go a long way with them. This is what the antennas look like for the tendrils. You can see them in the ground here. So these are flexible loop antennas. They have a diameter of 60 centimeters where they fully extended. So when snake goes in there within the antenna or crosses the antenna is 25 centimeters from it, it gets detected, and the time when it is there, gets recorded into the memory of the controller. And in the forest, we have the long cord antennas. So these are 20 meters long. It has to be a loop. So it's actually 40 meters long, but it has to be looped to record. And anywhere the snake crosses here or comes close to it is recorded. It's all solar powered. Leonard works very hard to keep these solar power panels going. The bears go there. There's an excellent picture of a bear. We're monitoring them with the remote cameras. That just was sent to us yesterday. I was going to put it here, but I didn't have time. So here there's 3 solar panels for each of the control boxes. So this runs the 3 lines. Here's some preliminary data. We just started this design in 2023. So these are should be, they just preliminary, these results. We hope to get more later on. So this is for Observatory Hill, showing the number of detection events along the edge, transitional and coniferous. The detection events are, the snake has to be gone for at least 5 minutes because we're recording every 2 seconds. So we have, our data sets are sometimes hundreds of thousands of detections. So we had to organize them in some way. And the days, that's the number of days, the orange column that the snake was detected at the particular reader. This is these are pooled data for the 4 replicates that we've got so far. So in total, we have 113 snakes been tagged since 2011, 26 since 2020. So those, at least those are still around. And we have 227 auto detection events on 121 snake days by 22 snakes since 2023. So this is really encouraging because we just putting these loops into natural habitat, we're not using ACOs on them. We have another system with ACOs on top. So from here, we can see that the edge and the transitional habitats are used most, but they also use the coniferous habitat, and this is somewhat unexpected, and we hope to gather more data on the use of the coniferous forest. It's not trivial from the conservation point of view, because we may need to also include these areas into the critical habitat. There's something else that it's really interesting that's come out of monitoring these snakes for 24 hours a day, is the time of the detection events by season. So the blue is spring, summer is orange, and the fall is the gray. So the time of the day at the bottom there, the 24-hour clock. So they start their activity around 8 o'clock in the evening and continue until about 4 a.m. So they're mostly nocturnal. And there isn't really that much of a difference between spring, summer, and fall. The fall, they start being active earlier, and it gets dark earlier. So this data can be teased a little bit more to show when it gets dark. Well, we've been using traditionally artificial cover objects. And we do have 2 systems that where we had, when we doing a pilot study, where we had these tendril antennas, automatic recorders, and we have ACOs on artificial cover objects on top of them. So, so now we can compare what happens when we have ACOs on top, these artificial thermoregulation spots, and when they just move into the natural habitat. So here we have the blue ones are the data that I just showed you with no ACOs on top, just the automatic recorders. And the orange ones are from these data with the ACOs on top. And what you see, you get lots more data and activity if you if you provide them with thermoregulation sites. So they do have to still thermoregulate. but there is a nocturnal activity and corresponding more nocturnal or evening activity when there are no ACOs. You can look at this in the, oh, that was spring. This is summer. And there's not really much difference. So the difference was in, let me go back just here. So in the spring, this is the difference that we got. What about in the summer? And I already gave that away. Not much of a difference. They mostly nocturnal, little bit of daytime activity. So what did we learn about that? If you put an ACO on top there, you're measuring different and complementary aspects of activity when compared to how they move in the natural habitat. So it's sort of obvious that ACOs, you enhance in thermoregulation, you're catching thermoregulating snakes. And when we don't have the ACOs, we're getting the foraging, seasonal and daily movements, and possibly dispersal with the loop antennas. So if we only use ACOs, we get an incomplete picture of the activity and habitat use. And once we get more detections, which we hope to do in the near future, we can start relating activity to habitat use and examine this in more detail, break it down more. So I'm just very quickly going to show us, we have Hills Rifle Range is another site. We have 2 mini setups like this. It was very difficult to get the units up there, but fortunately the Air Force came to our aid, and they brought the huge Cyclone helicopter to bring our stuff up on top there. We don't have very much data to date, and it's set up somewhat differently. So what our next steps are to tag more snakes, and that's Rebecca's working very hard on that, and continue monitoring these systems to get more detections and better representation of what the snakes are actually doing. And we want to set up the Hills Rifle Range if we can get some more units up there, so that is our replica site. And thank you so much to our funders, field crew, observatory health staff, like they've been excellent. And of course, the auto reader equipment, and Eric and Leanne and the Sharptail Snake Recovery team. Otherwise, we couldn't do that. Like these units are extremely expensive. So we so appreciate their help and to be able to do that. Thank you.
Awesome. We have time for 1 or 2 questions.
Thank you. That was amazing as always. Can you tell us a little bit about the approximate costs of setting up a system like that?
Wow.
Sorry.
I'm not entirely sure, but it's the tens of thousands of dollars. So it's expensive. If you just want the portable, the handheld units that Rebecca was talking about earlier, it's very tedious, but they do work too. So they used to be about 4,000 or so. So they're manageable. But to get in those boxes, and it's also changing all the time, the costs. So I think Eric was able to get them at a reduced rate because we good customers. Christian wants to say something.
I think the cost is about 20,000 per setup.
Okay. Thank you.
Can you tell us a little more about how you decided on the placement of all the antennas? Because Observatory Hill isn't super small, so how can you possibly cover such a big area?
Well, I tell you, it's not random. We started out in places where we had pit snakes already, where we put this setup. And it got difficult because we had to find somewhere where we could, it's very steep in places, so where we could have the edge, the transitional, and the closed forest. And they turned out to be, like everything, when you go to the field, everything is slightly different, so your replicates are slightly different. We started out with the idea that we put a sort of a long line, maybe 100-meter transect that we select where these, where we have pit tag snakes, and then select a random point along that as our center for the transect and go from there. We had to shift it a little bit because you encounter these steep drops and stuff, like you don't want to lose any field assistance there. So it's sort of, yeah, it's not random, unfortunately. Mark.
Yeah, I'm just curious to know. I know that I tried working with a reader that was based on a design that Cynthia Pazowski's lab had done, and we were trying to get into the depth of the soil for long-tailed salamanders. It was very challenging to carry this big piece of equipment out, but I was just curious to know if, in addition to the passive, are you also doing active reading, and if there's ability to do that for like at denning sites and to locate, and if that would be of any utility?
Yes, yeah, they read through the rock, and so that's why we can find them with those the portable readers when they're underground. We rarely see them. So that's, and we've done some experiments, Kristiina and I did at one point, long time ago. So it does go through rock as well. And we have found at least one hibernation site where multiple animals go back. The hibernation sites are scattered across the hill, but some animals go back, and they don't go very deep. So you can even find them during January or December.
We're doing pretty well. The other room is a little bit behind us. We do have time for one or two more questions if we have any more for Kristiina. Great.
Great talk. So this is kind of just stepping out of this pit tagging stuff, but in terms of the range of sharp-tailed snakes, do we think because they're so cryptic and sneaky that there might be other locations, like on Vancouver Island, the Gulf Islands, where they might be, and we just haven't found them yet? Or
definitely,
yeah.
There's one site. There's a historical site from the 80s on Galiano Island, and Laura Mathias just, people have been looking, and just this year confirmed that, yes, they are still there. There has been some new localities. I just trying to go all the way back to the beginning here to show the map. Sorry about this. There may be a better way to do that, but I can't focus there now. Okay, we get into the map. Okay, here's the map. Las Kitty Island is last year. So that extended the range quite a bit. And we also have new occurrences near the known ones. Yeah, they almost certainly there are others. The Pemberton is still isolated, and it's a totally separate population. So this genetic work is going on. We've been collecting blood samples, with Leanne has kindly organized that they sent to Steve Lauhit and his students, and they just done a little bit of preliminary analysis, and the Hills rifle range and the observatory hill studies, they group together. They in separate groups. So even within that short distance, there is see the blue and the red dots. These are hills, and then they are isolated because there's roads and different habitat, but they're not far apart as the crow flies. And also they found that even on Observatory Hill, within small distances, there are genetic groups. So they very fractured genetically, these populations. So now what we need to do is get some samples from the Gulf Islands and also really important from the Pemberton area.
Awesome, thank you. I did see one more hand at the back, so we'll do one more before we break.
Okay. So I'm not sure if this question is better directed towards Rebecca or you, but I'm just wondering, what is the furthest distance you've actually found an individual sharp-tailed snake to move? Like this
Rebecca.
might have been covered. Okay.
Say it again. It was 282 meters, straight line, and that was in one area in Observatory Hill, and that was with 96 meters of elevation gain. So that's like a pretty big distance. Do you remember? It's a couple
Yes.
months, right?
Some of them are years, like the home range maps, I don't know what to call them anymore, the ranges, but some of them are for 10 years or 15 years, and over that period, they do not move very much, but there are a few that sort of are caught on 2 of our units, for instance. Very sedentary, but the occasional long-term, 200 meters for us is long-term.
That's pretty
It occasionally does occur.
All right, thank you very much, Kristiina.